Hi, boys and girls. We're doing a kind of a short notice impromptu stream with rational empiricists on variation and information. And we have a paper that you wanted to start with, right?
Yeah, basically.
Okay. So I do believe we're live. Yep, we are live. Oh, that would be a good thing.
Darn it, speed, we're late by 15 nanoseconds.
15
nanoseconds.
Actually, 4 minutes.
Oh.
Yeah, I think
how many nanoseconds is that?
That's quite a few nanoseconds. That's a few billion nanoseconds, like about 4 of them. We're really bad people. Okay, so is it genetic evolution? This is the one I want. Okay, and we'll try to make it full screen. So what part did you want? Did you want to go through the abstract?
Let's see. Yeah, I think we should go for the abstract first.
Okay. So let's see. This is a paper from 2008 by Stern and Orgagoso. Orgagoso. Wow. Virginia Orgagoso. Okay. I bet she has trouble with that at the grocery store. Okay. So you want to go ahead and read, or you want to do the abstract?
Sure. Yeah, I kind of wanted to talk about it a little bit first because I was basically looking into some of my past emails about from the professors, of course, about some of the relative significance of coding versus non-coding changes. Specifically in this paper, they call them cis-regulatory changes. And so kind of looking into this idea again of, okay, we know that both occur, but what is the relative significance or usage of either in terms of morphological evolution? Because I want to keep it at, or I think the main discussion is basically morphological evolution, because that's kind of what the creationists always harp on, isn't it?
Yeah, but they always ask that question. No one ever answers that question, and any attempt to answer that question is usually met with shifting the goalposts. So, but yeah, that is the key to understanding evolution, is how we went from fins to limbs and things like that. So,
right. Yeah, and that's this idea of, because we know that evolution happens via variation in populations. And I guess the question we're trying to answer at this point is there a leaning of one towards one in terms of the relative contribution of genetic variation in a population? And so that's what this paper is basically going over, is basically talking about some of the arguments for the cis-regulatory hypothesis, they call it. And then also kind of seeing whether those arguments work. I feel like they eventually come to the position that that it's not as strong an argument as empirically as its proponents would say. I think specifically the cis-regulatory hypothesis is talking about most of morphological evolutionary changes being regulatory, right?
Yeah. And cis-regulatory is like something within the neighborhood. Right. And and I've heard it used even as within the whole chromosome, and transregulatory as, there being something in another chromosome that gets transcribed, like an RNA that, comes and affects something over here. But is that your understanding of cis-regulatory? I think it's even in the neighborhood, like, in gene packages, like little groups. But
Yeah, I think so.
yeah, and we'll see if they define it. But we might read the abstract and see.
Okay.
But I'll let you do that.
Okay.
Is that okay?
Yeah. Do you want me to read the whole thing or a specific part of it, or?
Just the whole abstract. May as well read it out loud and see what we say.
All right. So is genetic evolution predictable? Evolutionary developmental biologists have argued that, at least for morphological traits, the answer is a resounding yes. Most mutations causing morphological variation are expected to reside in the cis-regulatory rather than the coding regions of developmental genes. This cis-regulatory hypothesis has recently come under attack. In this review, we first describe and critique the arguments that have been proposed in support of the cis-regulatory hypothesis. We then test the empirical support for the cis-regulatory hypothesis with a comprehensive survey of mutations responsible for phenotypic evolution in multicellular organisms. Cis-regulatory mutations currently represent approximately, 22% of 300. Is that a one or free? I can't tell.
331.
331 identified genetic changes, although the number of cis-regulatory changes published annually is rapidly increasing. Above the species level, cis-regulatory mutations altering morphology are more common than coding changes. Also above the species level, cis-regulatory mutations predominate for genes not involved in terminal differentiation. These patterns imply that the simple question, do coding or cis-regulatory mutations cause more phenotypic evolution, hides more interesting phenomena. Evolution in different kinds of populations and over different durations may result in selection of different kinds of mutations. Predicting the genetic basis of evolution requires a comprehensive synthesis of molecular developmental biology and population genetics.
Okay. I am going to put this in the tags. Okay. So where do you want to go from here? Let's see. I guess I'm clipping a little bit.
Yeah.
Can you read that okay?
Yeah, I can read it.
Okay.
Let's see. There was a specific figure. Like it just caught my attention, I think.
The figure?
Or yeah, there we go.
Okay. Let's see. So what do we got here? We got AG, we got introns. Go ahead. Do you know more about this than I do? Because I'm just looking at it. In answer 1, answer 2, we got the Tata box. Or Tata. Tata is a consulting company in India. So I like to say Tata box. I don't know. It's up to you.
Right. Yeah, I think what it's showing here is you got your cis-regulatory regions and you have your coding regions. Coding mutations affect that part, and regulatory mutations affect the other parts of it.
The enhancers.
Yeah, the enhancers.
Yeah. Okay.
Let's see. All right, so maybe scroll down.
Okay.
See it. If something catches your attention, you can. I haven't reviewed this one in a while, but I found it interesting when I did look at it.
Definitions. Let's see where they start out here. At the beginning. Let's see. Let's see what they got to say. Natural selection causes predictable changes in phenotypic variation. Okay, this predictability is 2 levels. First, quantitative. Quantitative genetics provides predictions for short-term response to selection, given estimates of heritability. So how do they tackle this problem is what I'd like to know. These are probabilistic predictions. And due to historical contingency, which means what monophyly, what you got is what you get to work with.
Right.
Nonetheless, in some cases, precise predictions at the phenotypic level have been fulfilled by observation. Okay.
So I think below this or on a different page they mention non-synonymous mutations somewhere. Let's see.
Non-synonymous. 10. Oh, whoops.
It's somewhere below.
Nope. Unless they do one word. Yeah, here we go. Okay, so they spell it with one word. So this is the first instance of non-synonymous that we have.
Okay, there we go. Yeah, that's the first page I want to go over, basically.
Okay.
Let's see. I can. Let's see. Can I read this? I'm not sure.
Too convinced? Do you want to start there, or do you want to? Let's see. Regulation.
Yeah. That sounds good.
Okay. Are you going ahead?
Oh, well, some of the is kind of.
Genetic evolution is predictable in at least some general sense. We point out that there's already an uncontroversial general theory of genetic evolution. Non-synonymous mutations are predicted to contribute more to phenotypic evolution than synonymous. Yes, I would go along with that. Unless, of course, we're dealing with those enhancers. And then non-synonymous and synonymous don't make sense for those.
Right.
There's, of course, a good reason for this prediction. Non-synonymous mutations alter the amino acid sequence, likely to affect, the proteins. Okay, in contrast, synonymous mutations do not alter the amino acid sequence. They can modify gene function through other mechanisms, through changes in the translational efficiency. Of course, there we run into ribosomal pausing and that possibility. In addition, there is empirical evidence that non-synonymous mutations have contributed more to phenotypic evolution than synonymous mutations. Yes, we would guess that, because it's going to change the protein shape. But I think what they're getting at overall in this paper is that if you change a protein, you might change a protein, like you might change a calcium or a cartilage producing protein in such a way that it would start to exude minerals and become a hard or a bone. So you can go from cartilage to bone with, like a gene duplication and some mutations, right? You could end up with a different kind of cell. So so that would be the non-synonymous mutations making an effect on the protein. And then the other effect, which really is at play in the embryo, is when an embryo develops, there are 3 spatial dimensions and also a time dimension. So it's a 4-dimensional development, right?
Right.
And if you change, like you have one small mutation in one of those enhancers, then that gene may be transcribed more or less than another one. So when you go, like if you have a cell on the posterior portion or anterior portion of the zygote, okay, it's going to be exuding, is that the word? This protein, say the protein ends up out in the intracellular space. Okay. And if there is less of it, it is going to be a lesser gradient than if there's more of it. Okay. So it's going to affect the shape of that organism because of the way it spreads out across that zygote, that sort of thing. So you're dealing with the spatial thing there. And then the other thing is, as cells differentiate, they go through this complicated maneuver of different pathways, so that a mutation may cause a gene to switch off entirely, so you don't have the protein, or switch full on, so you got a lot of the protein, and it's going to evolve into a different kind of tissue. So without any change in the protein at all, we could end up with a change in morphology. Right. Like for instance, your tibia could be much longer because something is available longer. It's the concentration sticks around for a longer period of time, and hence, the cells keep dividing and keep pushing outward and growing a longer limb. So you could end up going from a limb bud to a lengthy limb, something that sticks out, and all of a sudden you get a different kind of animal. So,
so I guess the next thing we might want to ask would be, because they're talking about, I believe, or they mentioned coding changes to developmental genes specifically, because I feel like that makes sense, because like if you're changing the down-regulated genes, if you're introducing coding changes to them, it's not gonna be as broad a change. Like you know what I'm saying? Like it's not gonna be as emergent basically as a larger, or not a larger change, but like a change more upstream the regulation chain basically.
Yeah, I think regulation can change morphology. It seems to me easier than a protein coding change.
Right.
Because then you're gonna,
Oh, go ahead.
And it's less likely to be selected against too, because it's less likely to create something. It's going to create variation rather than stronger alterations like you would get with a if you change the protein entirely and the protein no longer did what it no longer created minerals or mineralization, right? You could completely change from a bone to a cartilage, that sort of thing. So that might be negatively selected, whereas a regulatory change may change shape because it's going to change that four-dimensional spatio-temporal, what would you call it? It's almost like, you familiar with analog and digital?
Vaguely.
Okay, digital changes are either on or off. Something is either on or off. But analog has grades from 0 to 100. To 0 to 100%. So it could change that, and then that mix across this really complicated dimensional state space that you have for this cell could modify function much easier, I think, rather than kill the thing or completely change the type of tissue, right?
Right. Yeah, that's a good point. Basically, that a regulatory change doesn't have to switch it on or switch it off. Basically, it's a gradient of change. Basically, I think.
Yeah, Mutable made a point here.
Oh, is he Mutable in here?
No, he's in chat. He's probably walking. Yeah, he's probably in the shower. And texting and showering is illegal, isn't it?
Maybe. I thought it was more dangerous.
Oh, yeah, especially if you have a 220-volt phone. If you have one of those really neat lithium batteries, or maybe an iPhone, I think it can kill you.
Yeah.
It's dangerous to the phone anyway. Okay.
Yeah.
Not always solid.
Let's see. So I think later in the paper they talk about this one argument for the cis-regulatory hypothesis, basically, and how, or whether or not developmental genes are as pleiotropic as proponents of it say. Because, like, if you have a pleiotropic gene, then changing the coding sequence of that is gonna have a bigger effect than changing the regulation, right?
Okay. Would that be under arguments for cis-regulatory hypothesis?
Yeah, I think so. Yeah, I'm not sure exactly how it's organized, so we may be going back and forth.
Yeah. Okay.
So maybe just scroll down and I might see it slowly.
Oh, they're talking about Escherichia coli or E. coli. I would say coli instead of coli. Certain gene products, what we now call transcription factors, bind to specific nucleotides, adjust. Okay, that's binding to those.
Right. That's the basics of it.
Binding to the enhancers. Okay. And let's see, conservation of coding. Where was I?
I think it was right there.
Was that right
Yeah.
there?
Or that's where
Oh.
where you left off.
I'm not entirely sure where I went. Oh, no, I was right here. I was at the bottom of the page. Okay. Oh, Monad and Jacob. Those guys are lots of papers by those guys. Enzyme act, and they're mentioned in a lot of books, certain gene products, even textbooks. Will be alcohol. Okay. Over the past 30 years,
maybe search for a pleiotropy or something.
Okay.
See if we can narrow it down.
Here we go. So there's the first occurrence of pleiotropy. Right here.
There we go. The population genetics argument. Let's
okay.
That.
You want to read?
And just to be clear, we're not arguing with this paper. We're trying to learn from it, right?
Yep. Yeah.
Because we're not scientists.
Yeah, we haven't checked more recent work to see how this paper's been received.
Yeah.
That's something that I really need to work on learning how to do, is finding out where this paper was cited and finding critiques of papers, because oftentimes somebody will put a paper out and say, oh, this supports my hypothesis. But then if you look at all the places that it was cited, you'll find that either it was accepted or it was not accepted. And I ran into that problem on Standing for Truth the other day, where he had put out a paper about it was about recombination and mtDNA. And did I talk about this live, or were we backstage when we talked about this? I can't remember. I'll repeat myself. And it turned out that the paper he referred to used invalid or dodgy PCR techniques. And the paper was from like 2008 or something like that. And it turned out that later on, the paper was not well received at all. They weren't able to reproduce this information. So it's really important to look ahead after you buy into one paper, to go ahead and try to see if anybody's mad at those guys.
Basically.
Yeah. See if they pissed anybody off.
Yeah.
Okay. You want to go ahead and read and paraphrase or whatever? Read, paraphrase, or do what you like.
Let's see. This argument, or the argument, is that natural selection favors cis-regulatory mutations because they have fewer pleiotropic effects than coding mutations.
Okay.
Let's see. Like I said, I can barely make it out. Some of the letters aren't showing up for me.
Oh,
just second reading.
No, it's sometimes YouTube does that, and so other times it doesn't. If you reshare it or whatever, maybe I can make it a little bit bigger, but I'm not sure. If I make it bigger,
there we go.
Then yeah, we can do this, and we can read one column at a time. Okay.
Yeah, that'll work. Okay. All right. This argument has two core assumptions. The first assumption is that natural selection should favor mutations with fewer pleiotropic effects over those with more pleiotropic effects. The second assumption is that mutations in cis-regulatory regions should have relatively specific, less pleiotropic effects than mutations in coding regions. We first explore both assumptions, which leads to support for this line of reasoning. Then we critique this argument.
Okay, and let me reposition. Go back up to the top. Now, pleiotropic would be where the protein has an effect in different parts of the organism. So it's involved in more than one pathway. Is that a good way to put it? And what they're saying, but then, okay, if the gene is expressed and there's only one gene, if there's a change in the regulatory portion, it's still going to have pleiotropic effects. It's still going to affect how much of that gene is produced. So I don't know what they're saying here exactly. But go ahead.
or I guess maybe scroll down to that part of the argument. I know I'm jumping from place to place here.
Yeah, let's concentrate on this part. Okay, those
Okay.
pleiotropy impact evolution. The consequences of pleiotropy for evolution have been explored within several frameworks. Fisher's geometric model of adaptation, whatever. Let's say a framework in which a total selection acting on an allele is the sum of positive direct effects and positive or negative pleiotropic effects. Okay, and a framework in which so-called hidden pleiotropy. That's a nice little thing to highlight. Okay. Despite this variety of approaches, all models agree on several results. First, as the degree of pleiotropy increases, the rate of adaptive evolution decreases dramatically. That's an interesting comment. Why would that be?
Well, I think it's because if it affects more than one trait, then there's a good chance that affecting one trait might increase fitness, but affecting another trait might decrease fitness.
Yeah, so that would be like hidden pleiotropy, maybe.
Yeah.
Okay, this reduction in the rate of adaptation scales as n to the -1, where n equals the number of phenotypic dimensions along which the mutations can move the phenotype. So every one of the pleiotropic effects would be another dimension that could be changed, and the more of those there are, the smaller you're going to end up with affecting the organism, because most of them are going to be selected against, I guess, is what they're looking for.
Yeah.
Let's see. Where n equals to the number of phenotypic dimensions. Okay, because the universal. Okay, well, Waxman show that this result is robust to a wide range of parameter values and to alterations of the model.
Yeah, some of this math we may not be able to understand.
Yep. They show that the reduction in the rate of adaptation is probably even faster than n-1. So it's even worse than we thought. Let's see. So modifying pleiotropic genes is considered a bad thing. And then, of course, we have Haldane's dilemma.
Oh, yeah.
Which we hear about a lot. They love that one. Thus, okay. In contrast, the probability of fixation for an advantageous mutation without pleiotropic effects approximately 2s. Thus, strong pleiotropy will prevent fixation of most potentially adaptive mutations. So pleiotropy bad. That's kind of what they're saying. Don't do pleiotropy. Pleiotropy bad. It can be problematic. Unless, of course, it adds up the other way. It ends up beneficial on two different dimensions or more than one dimension. And then we could have a sweep and a massive change, and maybe even saltatory evolution. I mean, it's possible, yeah.
Right.
Okay, in real species where one. Okay, where do you want to go from here?
Let's see. I'm reading the last sentence on that page, so maybe scroll down a little bit. I guess we'll shift the column again.
But complex organisms can evolve quickly, both in the laboratory and in field. Either the models are wrong or the assumptions are wrong, or both. Interesting. Okay. So we're saying, in spite of all that we know and all that we've proved mathematically, the goddamn life happens anyway.
Right.
Okay. Okay, so now we're on the next page.
Right. All right, let's see. Given the diverse modeling approaches that have been taken, it seems unlikely that the general results are flawed. It seems more likely that the assumptions are flawed because organisms are, rather obviously, complex. It's unlikely that there is a problem with the assumption that organisms must adapt along multiple phenotypic axes simultaneously. Let's see.
Let's see. James Downard is in chat, and he asked if this was a different paper by Charles Fox. And no, this is not that paper, James. It's the paper I just posted in chat, and it's also in the description. Okay. I'm sorry, Andrew.
It's okay. Yes, some of this I don't think I'm old enough to understand yet.
I'm not old enough to understand it, so we're fucked.
If we can have James look into this, maybe he can come in and explain something.
I'd have to bring him in on Zoom, and I'm not sure if I'm ready for that tonight, but I guess we could. Charles Fox et al. Experimental evolution of the genetic load and its implications. No, this is more about the difference between regulatory changes and basically non-synonymous changes to the gene. And this is what they're going on about. Okay.
So maybe let's see if we find something interesting if we go down a little bit.
Cost of complexity is offered by results.
There we go. Gene pleiotropy.
Okay, where are they? Where are you seeing this?
Near the last paragraph on that column.
Right. Gene pleiotropy. Okay, there you go. The effect of an allelic variant on multiple different phenotypes rather than just one. Historically been considered universal, at least very common, by Haldane. I don't know. Since the gene exists in every cell of the body, it may be expected to affect the organism as a whole if it is expressed. Now, if it's methylated for some reason or wrapped up in a histone, it may not have any effect at all. So I think, Haldane 1932. So consider the source.
Right.
I don't know. I don't even think they knew about all the things that were wrapping up genes back then. So I don't think you could make a general statement like that. Okay, Sewell Wright in 1968 says the available evidence indicates that pleiotropy is virtually universal. But that was in 1968. This idea has been repeated. Okay, are they pushing back against that? Largely on observation of phenotypic effects of individual mutations and domesticated.
All right, so it says down here basically that these are observations based on largely domesticated and laboratory strains and races. And then it says recent studies have confirmed the existence of widespread pleiotropy throughout the. And then
working on it. Okay, genome.
The genome, for example. Let's see, examination of 501. You're gonna have to help me out with some of this because I'm gonna be reading it, but I need you to help interpret it, some of it.
If I can.
Let's see, examination of 501 morphological phenotypes in each of 4,710 deletion mutants of non-essential genes in yeast found that about 35% of deletion mutants affected at least two morphological characters. A second study essayed relative fitness of these deletion mutations in 21 adverse growth conditions versus a control medium. About 58% of genes displayed growth reduction in two or more conditions, which the authors considered to reflect pleiotropic effects in different growth conditions. These two studies have explored a limited number of growth conditions and aspects of morphology. In addition, yeast possess far fewer phenotypic features than multicellular eukaryotes. Therefore, deletion of genes in more complex eukaryotes is likely to cause at least as much pleiotropy as observed in yeast. Although gene pleiotropy may be relatively high in yeast because they have relatively few genes, it seems likely that the classical view of widespread gene pleiotropy is accurate.
Yeah. I don't know if I agree with that. So.
Yeah. I guess let's grant that
for yeast anyway. We'll grant that for yeast.
Yeah,
they don't have that many tissue types.
Right.
One tissue type. Further analysis of these data sets. Okay, you can go ahead. Let me know where you want to go. Pleiotropy not correlate. Okay, so they go through. Okay, so they're going through a bunch of evidence for the idea.
Right. Yeah, so I think it seems like pleiotropy seems pretty common.
Yep.
There, they even say it. Okay.
Pleiotropy genes
versus pleiotropy genes.
What? It is a striking fact. None of the population genetic models exploring the effects of pleiotropy have incorporated realistic models of gene structure and function. All have assumed that gene pleiotropy is universal. And more important, so I think they're pushing back again against this idea.
Let's see. Donald Kronos in the chat says he can explain why pleiotropy is so common. And he says he sent you a text message. Should we let him in? Since Immutable doesn't seem to be joining. Still there? You there, Speed? Might be muted. I can't hear you.
Oh, I was muted. I'm going to go try to find my phone. Do you want to go ahead and read this part or not?
Sure. Yeah.
Okay.
Speed. Someone we know is in the chat.
Oh, not that guy. Oh my God. Should we block him? I don't think we can. He's a mod.
I think that would be too mean.
He's a mod.
I want to be the better person. Let's see. In most cases, each of these cis-regulatory modules acts independently of the others. That is, these modules can be experimentally dissected from their native genomic region, artificially coupled to a heterologous promoter and reporter gene, and shown to drive reporter gene expression in a small part of the complete pattern. Each cis-regulatory module is a collection of transcription factor binding sites that, together, encode a transcriptional output. Usually each module contains multiple binding sites for each of several transcription factors. Okay, that makes sense.
Okay. I'm back.
They appear to evolve as structural features of the genome that are independent of one another. The simplest observation of supporting this contention is that these modules are often separated from each other by large DNA regions, thousands to tens of thousands of base pairs long. In addition, although the modules can retain conserved function, the DNA regions separating them often evolve in size. So I guess they're saying there that the space in between, like you're basically piling it up, basically, I think is what they're saying.
Yeah.
All right. Let's see. This is consistent with the current mechanistic view that the intervening DNA is looped out, allowing the transcription factors bound to the DNA to make contact with the basal transcription apparatus. This looping apparently enables cis-regulatory modules to reside very far, sometimes over 100,000 base pairs from the basal promoter. The modules can reside 5' or 3' of the coding region in introns or, in rare cases, in exons. Let's see.
Wow. Okay. Express in various tissues often contained.
Okay, that makes sense.
Well, we'll go to the next part.
Okay.
Up here. Small part of the total. Coding mutations may either eliminate gene function or alter the gene product. Modifying a single domain of a protein or an RNA molecule is likely to affect the molecular properties of this molecule in many of the cellular contexts. Now, that's an important feature right there. And that plays into the paper we're going to start going over tomorrow. Cellular context. If the gene encodes a transcription factor, then this change can alter the transcription of many other genes in every cell where the transcription factor is.
Okay, so that's the developmental gene context we're talking about there.
Yeah, okay. In contrast, mutations in cis-regulatory modules will normally alter gene expression in only a small part of the complete expression pattern. Okay, so they're saying that mutations are going to affect it in every cell context, whereas cis-regulatory will only could affect it much more specifically.
Right, that's the argument that's being put forward that they're trying to address, I guess.
Yeah, are they for it or against it? We haven't figured that out yet.
I'm not sure exactly.
Okay, regions are usually more modular than coding regions because cis-regulatory modules are more independent of each other than the domains composing a protein or RNA molecules. Mutations in these are likely to have fewer pleiotropic effects, which I would agree with that so far. This argument thus, but I'm not entirely, I can see where they may go against that. This argument thus supports the narrow cis-regulatory hypothesis that phenotypic evolution should result more from cis-regulatory coding changes. So that is the hypothesis. Okay.
Right. Yeah. So here they're basically saying that according to that hypothesis, that cis-regulatory mutations are most of the changes, is more supported in this case than the broad cis-regulatory hypothesis that says both kinds of changes are important. I think is what it's saying.
Standing is over here poaching.
Uh-oh.
trying to poach discussions for his channel, his growing channel. I think that's the only reason he stops over, is to poach our people.
Probably.
You poacher. What do we do with poachers? We hang them.
Oh, okay. I don't obligate violence here.
Okay. Well, okay, we'll just put them in jail.
Yeah, that's better.
Until we hang them.
Speed.
I know, I'm a bad person. Okay. So.
All right, difficulties with the argument. Okay, so now they're going to address they're going to see if it stands up to criticism, I think.
Yep.
For genes with pleiotropic effects or roles in development and in possession of modular cis-regulatory regions, this is simply the simplest way to conceptualize the problem and is consistent with the observation that many genes contain modular cis-regulatory regions. However, many genes may not have cis-regulatory regions organized into independent modules, and it is not yet clear whether mutations in non-modular cis-regulatory regions have non-pleiotropic effects. Although it is true that the majority of genes that have been examined in detail appear to possess at least some modularity in their regulatory regions, it is also true that this modularity does not appear to be always complete. For example, experimental dissection of the cis-regulatory region of the runt gene in D. melanogaster has identified a greater than Te-Kb region required for embryonic expression in 7 stripes. It was not possible, however, to dissect this region into smaller, independently acting modules. The transcription factor binding sites driving expression in 7 stripes may be dispersed throughout this region rather than being compacted into individual modules.
Okay.
Let's see. The file drawer effect may have caused an excess of modular cis-regulatory regions to be reported in the literature. Several of our colleagues have informed us of their unpublished attempts to dissect cis-regulatory regions.
Oh, I'm sorry.
Okay. That have not uncovered simple modular regulatory regions. These studies normally remain in the file drawer for an exception. See this one. Because most such studies have not been published, it is currently impossible to accurately estimate the proportion of genes containing independent regulatory modules. Okay, so I think they're basically saying there's not enough evidence, basically.
Yep.
That there may be some statistical effects.
So cis-regulatory modularity. Somehow I'm missing what it is. Modularity. That it's all in one group rather than scattered throughout.
Right. Yeah. It's that they're independent of one another. Basically, that changing one doesn't influence the other.
Yep. Okay.
Okay. I guess should we keep going or what do you want to do?
Okay, so Donald says he can help us, but and we've turned down his help. But could you put it in chat, Donald? Could you put something in chat for us? Because we got a lot to get through here. Well, where do you want to go next? Let's see. Should we try going to the conclusion? And
sure, yeah, maybe that'll help.
Yeah, because there's a lot here. Okay,
yeah.
So let me make this smaller for the moment until we can figure out where they conclude. Prediction. Let's see, prediction 3. Oh my God.
Donald Kronos a biologist by any chance?
I don't think so. But I'm very interested and curious. I think it's module is in, let's see, and it's all in one group. Exactly like I said, I wouldn't call them modules. It could be taken wrong. Okay. Let's see here. How many predictions do they make? Prediction 2. Okay. Perspective. So this is a perspective. Prediction 1. Okay, let's see if the data. Okay, there's the data. Okay. So let's take a look at the predictions. Morphological evolution results from more cis-regulatory changes than physiological evolution.
All right. So I guess zoom in a little bit.
Oh, okay. Plus.
Yeah, maybe that'll help. We can go into the other arguments some other time.
Okay.
Let's see. The regulatory hypothesis has been applied traditionally to the evolution of morphology. Occasionally, the idea that physiological evolution might involve more coding changes than morphological evolution has been made explicit. Carroll, 2005. Hoekstra and Coyne claim that the cis-regulatory hypothesis should apply to all adaptation or not at all. As they argue, and we agree, the division between anatomy and physiology may be a misleading dichotomy. At the very least, there is not a clear boundary between genetic mechanisms generating morphology and physiology. Both result from activities of genes that are embedded within regulatory networks. Developmental processes are influenced by physiological processes and vice versa. For example, development can be regulated by steroid hormones, by sugar and lipid metabolism, whereas endocrine glands are formed through developmental mechanisms.
Okay.
The distinction between morphological and physiological evolution that has been largely implicit in discussions within the Evo-Devo biology community is that physiological traits tend to result from genes located at or near the terminal points of regulatory networks, whereas anatomy usually results from the activity of genes embedded deeper in the developmental network. Genes that act at or near the terminal points of regulatory networks, named differentiation gene batteries, represent genes expressed in differentiated tissues to fulfill cell type specific functions. These gene products build muscle cells, make skeletal biominerals, mediate synaptic transmission, etc. They do not regulate other genes, and they do not control the progressive formation of spatial patterns of gene expression that underlie development. They receive rather than generate developmental instructions. Even though they may be expressed in various tissues, their pleiotropic roles are more limited than gene products that function higher in the regulatory network. For example, a coding mutation in D. melanogaster gene forked, a gene involved in terminal differentiation of bristles, can cause every bristle to develop differently, but all in the same way. In contrast, a coding mutation in wingless, a signaling molecule involved in diverse regulatory networks in various tissues, will alter the development of segments, legs. I lost my segments, legs, genitalia, and eyes in Drosophila, all in different ways. Therefore, it should be more difficult to evolve a coding change in wingless that somehow provides an advantage than a coding change in forked. Because the position of a gene within a regulatory network is likely to influence its pleiotropic roles, we might thus predict that genes at terminal positions in regulatory cascades, members of differentiation gene batteries, should result from coding changes more often than genes embedded deeper within regulatory networks.
Yep.
Okay,
So
can
let's
you?
say so the terminal one. So the ones that would affect more than one trait, one phenotypic trait, would be more highly conserved than genes like forked, that have just one effect, like on your bristles, right? So according to Kunin, his definition of meaning in genes and information, he would say that the one that is more highly conserved is more meaningful, because you can look across different species or different generations and find that it's more highly conserved. So it has more of an effect. So something that has more than one trait, you would call more meaningful, and sort of have, well, the creationists would say it had more information, but Kunin he differentiates between the information itself and the meaning it has. Okay, did I make that clear?
Yeah.
Okay.
Let's see. Can you go up to the figure top left?
Oh, top left. Are you kidding me? Okay.
Let's see. I want to see what this was. Cumulative number of coding changes, regulatory mutations, and other types of mutations have been identified over time as a result as responsible for phenotypic evolution. The slope of cis-regulatory mutations has increased in recent years. Current discovery rate of regulatory mutations approximately equals the discovery rate of coding mutations. If this reflects the long-term trend, then we expect ultimately to observe approximately equal numbers of regulatory encoding mutations.
Okay.
Okay, that kind of makes sense that they would both contribute.
Yeah. So yeah. So there so this involves what we know, what we've researched, rather than what is theoretically possible. So
Right. Yeah.
okay.
Yeah. We need more data. Basically, we have lots of data so far, and it suggests a pattern, but we don't know exactly what that pattern is.
Let's see. Donald, was it on? It was on pleiotropy. I'm not entirely sure. So
Yeah, I think so.
he was lagging behind us a little bit in his chat, but I haven't been paying too much attention. Okay, so where are we?
Let's see. I finished reading the last part of the prediction, so let's see.
Okay.
Okay, so now they're talking about how to test it.
Yeah, first we examine traditional morphology versus physiology hypothesis by classifying. So, how did they examine this stuff? They go across a number of different studies.
Yeah, maybe. Or, okay, go back. What was their prediction again?
Hang on a second.
Sorry,
Here.
I'm making you move it back and forth.
You suck. Their prediction was way up here, and we gotta go that way. Okay.
Morphological evolution results from more changes.
For more cis-regulatory changes than physiological evolution. So, what are they calling physiological evolution exactly? Maybe they made that clear earlier. That would be like endocrine changes or paracrine things that
Probably
that affect extracellular changes, that sort of thing. They're calling that physiological.
That would make sense.
Yeah. Okay.
Let's see. Yeah, I think they're basically saying that it depends.
Yeah, I'm not sure.
Yeah, I think we would have to go into this more some other time.
Yep.
Yeah.
Well, let's just quickly look at their other predictions. Prediction 2, the strength of selection and the evolutionary timescale should influence the spectrum of evolutionary relevant mutations. Why is that totally meaningless to me at this point? Just went right through my brain and it came out the other ear. The strength of selection and evolutionary timescale should influence. That's prediction 2.
So I think they're saying it depends on selection as well.
Yeah. Well, obviously. I'm going to go with that. So where's prediction number 3? They get far into this stuff here. Am I missing? Oh, bristles. Oh, you love bristles. I see why you found this paper. Were you searching for bristles again? Were you Googling for bristles?
No.
No, you weren't? Okay, well, there you go. Bristles and Drosophila.
What a wonderful coincidence.
Oh, yeah. I don't fucking believe it's a coincidence. I think you're... Ooh, this is nice.
Uh-oh.
Nice.
Don't show speed regulatory pathways.
There's your scoots and shaven baby. That's a gene. Shaven baby.
Shaven baby.
Yeah, isn't
that these names?
Isn't that illegal?
Wait, you saw Scoot? The Scoot?
Yeah, Scoot on the right. Right there. Can you see Scoot? It's right.
I can't make it out. Can you zoom in a little bit?
Oh,
let's see. Is it on the? Is
it on the right?
The right? The right, right?
Scoot's good. There's your Scoot development. It's a Scoot development pathway. This is good. This paper should keep you entertained for a while.
Oh, there we go. I didn't see it in the middle there.
What did I do? Yeah, it's right there.
Huh.
There you go. Yeah, right in the gray area. Amazing how the mind will cut that out. Neuromusculine, quail, senseless, hindsight, couch potato, snail. I love how they name genes. Couch potato. If they knock out couch potato, the thing probably just lays around, doesn't do anything. It doesn't eat. And snail, it goes slower. Spineless. Spineless. They don't have spines. Wait a minute. Senseless. They wander around senselessly, kind of like me. So pentaplegic.
Pentaplegic.
That is 15, right? 15-fold, something like that. Decapenta. Doesn't that mean 15?
Iroquois complex.
That's an important developmental gene, actually. Wingless is too.
Okay.
Knots in delta have an effect on the posterior anterior development of an embryo. And neuromuscular, I have not seen that. Okay, this is a really cool diagram for development. Things that affect cell adhesion, and actin and microtubule distribution strongly affect. Like, okay, sometimes if you imagine some cells are embedded in like cotton, and if you were to drop a little bit of red ink in cotton, it would diffuse out to a certain extent, and these actin and microtubule meshworks exist in between cells, and they can slow up the diffusion of a protein or a growth factor. And that all affects development very heavily. And cell adhesion would be sometimes cells just, like a layer of cells will just break up and become mesenchymal, and it'll start to migrate and then form different areas and actually diverge into different organs and different parts. And other times they will stick together, so they'll be more sticky, and they will form a nice layer of tissue, or they will form like a clump or whatever. So there's all very nice, very, very nice, very nice paper. I wonder how far the paper goes. Yep, that's about it, I think.
Yeah.
I think that's kind of the end of it. But
So
we might want to read this paper and revisit it at some point. But there's a lot of interesting stuff here.
Yeah.
And also look at other papers that refer to this one and see what everybody thinks of what they did here. But I think overall, if you're trying to figure out whether or not it was a mutation to a gene or mutation to a cis-regulatory network, I don't see how from studies on yeast you could decide that for studies on higher vertebrates, or even what happened during the Cambrian explosion, because but if there is a lot of pleiotropy, and that's what they're saying, then those genes are going to be more highly conserved. They're going to be more meaningful. So if you're looking at monophyly, right? You're starting at a base and you're evolving into some different thing. Those more conserved regions are going to restrict what can happen, so that something cannot turn into something very, very different. It's going to be constricted, and that's because the conserved portions are more meaningful per kunin, that sort of thing. So anyway. So should we invite Donald in so he can explain what he was going to tell us?
Sure, yeah, and then I think we should.
Or do you want to wrap it? Oh, wait a minute. Look, conclusions.
Conclusions.
You want to read it?
Sure. Zoom in again.
What? God, you're needy.
I'm sorry. Let's see. In the absence of a population genetics framework, evolutionary developmental biologists have inferred from our current understanding of gene regulatory networks, 2, our understanding of gene structure and function, and 3, the extensive conservation of developmental genes, that mutations in the cis-regulatory regions of developmental patterning genes are likely to underlie most of phenotypic evolution. However, no single argument proposed thus so far provides definitive proof that cis-regulatory mutations constitute the predominant cause of phenotypic evolution. By considering development and population genetics simultaneously, a survey of published data suggests that patterns of genetic evolution are not entirely obscured by historical contingency. Population genetics and development must be considered simultaneously to make sense of the data. It may be unhelpful to pose the coding versus cis-regulatory debate as a quantitative question. Do coding changes explain more of phenotypic evolution than regulatory changes? It may be more productive to turn the question around and ask what kinds of phenotypic changes are expected from particular coding versus cis-regulatory changes in specific genes. As we show, patterns in the currently available data imply that morphological and physiological traits are caused by different frequencies of both. This is consistent with our molecular understanding of how coding and regulatory changes might influence physiology and morphology. Yeah, so I think they're basically saying it depends.
Yeah, and that's kind of what the whole paper says is, whoa, let's not go with the paradigm.
Right. We need to be cautious. Basically, we need to look into this more, which I think is my naive opinion. I think that's a good way of looking at it for now, at least,
until we get looking at everything.
Yeah.
Doesn't think paradigms can get us in trouble. Okay, Donald, turn into something very different.
All right. Bring off the paper.
Punctuated equilibrium. Okay, should I get rid of the paper?
Yeah, I want to see our faces again, or well,
Um,
our icons. There we go.
oh, you can actually see me. Boo! I'm here. I just put my tinfoil hat on for this one.
Uh-oh.
Anyway, that's a pretty cool paper. I really get excited about that diagram. That diagram had
Yeah.
a lot of the developmental genes in it, along with a bunch of others that I had never heard of, like couch potato. Have you heard of couch potato?
Nope.
Yeah. Anyway, so yeah, so basically we read the whole paper, and it's basically saying, think. It's basically telling us to rather than choose this one hypothesis, which seems to be dominant, that it has to be cis-regulatory, that we want to look at both, which is kind of what I thought going in, or at least they have not changed my mind. That was what I had my mind made up as in the beginning anyway. So
yeah.
Hey, so Dax here. Oh, hey, Donald. Sorry,
As far as I can
we
tell.
wanted to get through the paper.
Yeah, no worries.
If you have
Yeah,
too
I
many
just wasn't
people,
sure. I wasn't sure if you were seeing my question of could I help, or if you weren't seeing it at all or what. So
I was
just
seeing I saw about 1 out of every 10 chats. So
figured about that.
yeah.
Any rate, yeah, a lot of that actually is understandable without even understanding biology. And I'm not saying I don't understand biology. Biology is just a matter of biochemistry in large and small systems alike. But if you look at the interaction of biochemistry, if you look at the interaction of chemistry in general, any complex system, including of course any complex chemical system, if you change one piece of the chemistry in there, it's going to have a chain reaction that's going to affect everything in some small way. The question is how much and in what ways. So there's technically two.
Right?
Yeah, two questions there, and so every mutation, regardless, every gene mutation is going to have multiple effects, but at what level, and is it going to be noticeable?
Right.
Yeah, and even mutations that do not have, basically random mutations that are not involved in producing a protein that is used
non-coding.
in any function. So totally non-coding. It's still going to take resources. It's going to have an effect. It's going to take space.
Yeah, even if they don't change any protein, even if it is in a coding region, but it's a change that doesn't change which protein gets encoded, it still has an effect. It's just not having that effect. It's not having the effect of changing what protein gets coded.
And that is what the ENCODE project is all about. Okay, the ENCODE project wanted to find every gene that was transcribed, even a little bit. Anything that was transcribed, even a little bit, because those things overall can add up and create medical problems, especially for human beings, which live way past their reproductive usefulness. I mean, we should die after we've reproduced, and raised our children. Then we should go away and die, but we don't.
Here's the thing: any gene that can encode for a protein, probably will eventually. It's a question of how much. Generally, genes that encode for a protein, when they get so-called activated, they encode for that protein a lot. Yeah. And occasionally for another protein when errors are made, because that happens. Sometimes close enough gets made. And
then the paper that we're going over tomorrow, the is a cell machine, and the part we're going to go over also talks about, like, proteins have the idea, or the idea we have is that proteins fold into these particular structures of machines. Well, number one protein can fold into more than one structure, and one protein
and therefore more than one machine.
And 25% of proteins don't fold at all. They just float around sort of either globular or as long strings.
Yeah. Well, it depends on what you call folding. They have a shape. And generally, I think, as I understand it, when chemists talk about them folding, or when biochemists talk about them folding, generally they're talking about the shape that they have. So even stretched out is considered a fold. It's just a stretched out fold. But yeah, when they're compacting, when they're compactified, it makes a lot of the molecule unaccessible or basically inaccessible, as opposed to when it's all stretched out, it's not. And that's part of what I was saying earlier. If there's a stretch of DNA that's able to encode for some protein, sooner or later that stretch of DNA is going to be stretched out and not folded up somewhere in the process of the cell producing or dividing, not necessarily one cell, but maybe a descendant of that cell, maybe in the process of producing gametes or whatever the case might be. There's very likely to eventually be some point where that strand of DNA is completely exposed, and if it can produce a protein, it will.
Yeah.
So Donald, if you mind, I'd like to ask you a question and hear your opinion on this. I guess what creationists would call micro variation versus new information kind of argument. I'd like to hear your thoughts on that.
Well, variation and new information are two completely separate concepts. Variation is a current state of things. Now, if they're talking about variation accumulating or changes in the amount of or the type of variation, that's a different thing. Can you give me a specific question?
Right,
just make your own. Just make your own. Just think like them for a moment. Make one up.
Okay. That's gonna be hard, but okay.
You might straw man them a little bit, but that's okay. I can deal with the straw man one. And if anybody in the chat wants to throw in a better one, if somebody can point it out to me, I can't see the chat, but I'll try.
Okay. All right. Let's see. All right. So I think what they're talking about is, say you have this population of hypothetical horses. Okay.
Okay.
And there's variation in the, let's say the
phenotypes.
The structure of the hair or the length of the legs, for instance.
Okay, so we got leg length variation, mane length variation, things like that.
Yeah.
Okay.
So they're talking about that as variation, and then they go on to talk about new information as kind of, okay, you get a leg and then you get a wing from that. How do you do that? I think is what they're asking.
Well, the leg and the wing are not genetic information. If you're going to talk about new genetic information, that doesn't come in the form of a leg or a wing. It comes in the form of DNA or RNA in a particular sequence. So, for example, when if you want to talk about new information, if you have the number 0, that's information. If you make another one like it, that's more information. Now, they would argue, generally, from what I've seen, they would argue, oh, but that's not new information. That's just more of the same information. Okay, but here's the thing: if you have a 0 and it changes to a 1, that is new information, but you lost some information also. If you have a 0, and it gets duplicated, so now you have 0, 0, and one of those zeros gets changed into a 1, now you have more information than you had before.
Right. Yeah, if you look at GCTA, if it morphs into GC or GTTA, okay, you actually have, I think, less information because there are
in a sense. Yeah.
But when it comes to biology,
technically speaking, from an information systems point of view, technically speaking, that's the exact same amount of information. It's just different information.
And not only that, but from a biological perspective, that is definitely different information, new information.
Right. And it's
even,
definitely new.
Even if we change it to TTTT, it is still very, very different, and and what they do oftentimes is they confuse meaning, which is what happens with happens to the sequence downstream, right? And establishing that meaning. Well, we have a paper that we reviewed a long time ago and we never published it, by Kunin, who talks about meaning basically as that downstream effect. So in this paper,
meaning is something we give stuff.
Well, no,
we
defines
perceive meaning, so therefore we give meaning so that we can perceive the meaning. That's a human thing.
Now, Kunin has a physical definition of meaning that he arrives at.
What would that physical definition be? Do you happen to know?
Okay, yeah. Basically, if you look at, say, we're looking at fruit flies, a whole bunch of different species of fruit flies. We're looking at 100 different species, right? if one part of the genome in every one of those is identical, that means it is more highly conserved, and he calls that more meaningful, because that would mean that, say, this one gene has a pleiotropic effect. In other words, it affects more than one function, so that if you
I see.
modify that, right? So if you
So
modify
he's
it at
thinking if
all,
a gene shows up more times, it's because the gene is more valuable in some sense.
right? And if you look at the genome,
I would dispute that.
What's that?
I said I would dispute that.
I kind of like his
Because
definition.
well, let me say
almost seems
why I
like
would
it's
dispute
less
that.
meaningful.
It can be the case, but the reason why I dispute it is because if a gene that isn't doing anything positive or negative for a life form gets duplicated and it still doesn't do anything positive or negative, there's nothing preventing it from keeping that duplication, and there's nothing making it keep it either.
Yeah, but
A lot of
that's
that stuff is strictly a matter of genetic drift, and in that sense could be considered basically random.
yeah. But if you
And
look
you
at
can't and you can roll a dice a million times and get the same number every single time. It's not likely, but it can happen. So that's why I'm saying that, is there some tendency toward that? Yeah, probably,
but
but it's not consistent.
if you look at all vertebrates, you'll find that some genes are almost the same in every single one of those vertebrates, and that means it's highly conserved. So,
Oh yeah, no, that's a different story. If you're talking about those genes that are the same in a bunch of different things, a lot of those are early developmental genes. Like, for example, genes that like the Hox genes that set up basic body structure plans and things like that.
Metabolism.
Yeah, metabolism. We need our metabolism. If our metabolism breaks down, we're not surviving. If we have an offspring without metabolism that keeps it alive, it's not going to be a viable offspring. So
right.
yeah, and those genes, you can, if you get a duplicate of the gene, and if the system works with both copies of the gene, and there's nothing, it doesn't, it's not a fatal error to have two copies. One of those copies is then free to mutate and break down and not work anymore, and eventually get enough changes to get some other function.
Yeah.
But but at least one of them has to keep working, because it's needed in that particular system. Now, if something else happens to develop that can replace it in advance, if, for example, our mitochondria were to diverge and we ended up with two different kinds of mitochondria, and cool. One of them outcompeted the other one, the other mitochondria might just completely go away. Maybe the one that's closer to the original would just be gone, and the other one might be so different that we wouldn't call it mitochondria anymore. We'd call it something else, but technically it would be a descendant of mitochondria.
Yeah. Okay, so we have 4 people watching us, and there's 105 people watching Erica. So, Andrew, is there anything else you want that you want to go over, or?
I mean, there's probably a ton of stuff we could go over, but it depends on like how long you want the stream to go. So it's all up to you.
Yeah, we should probably
we could always continue it in another stream another time.
Yeah, the other thing is we should find that Kunin paper on information and in DNA, and also Andrew, I can give this paper more of a read, and you could too. But okay. I think we pretty much gleaned what they were saying.
I did want to, if you don't mind, bring up something in linguistics that, depending on your knowledge of French, may or may not make any sense to you, but it's in relation to something that was said a couple of days ago. What's that?
All I know is that we don't like the French. That's all I know.
Oh, yeah. Well, you would understand me if I said parlez-vous français, correct? More or less, you'd know what I'm saying or asking. Yeah. Parlez-vous français?
Yeah.
Okay. If I say parlez-vous français, how do you suppose the word vous would be spelled? Can you guess a spelling for that word?
VU.
It's VOUS.
Oh, okay. I should have known that. VOUS.
Now, here's the thing: the S on the end isn't pronounced.
Right.
But what about the word parler? If I say je parle or vous parlez,
Yeah.
okay,
Parle.
yeah, is it? Or is it PARLES or PARLEZ or PARLET or PARLER? Because guess what? All of those are pronounced parler, unless the word following them starts with a vowel. Then you pronounce that final consonant. So someone was asking about, sort of one-to-one correspondence in coding. The written French and the spoken French are not a one-to-one correspondence of each other. The spoken word parler can mean a whole bunch of things that are spelled differently.
Yeah.
And you can lose a little bit of that information if it isn't in enough context in spoken form to be able to figure out which one is meant. Yep. In the written form, it's written down as part of the word. In the spoken form, that part is left out when you leave it alone. If you pronounce the word by itself, that final consonant gets dropped phonetically.
So what does parlay mean? Do or
speak?
Oh, speak. Okay. Okay.
Yeah,
and
PARLES, for example, is the second person informal. PARLEZ is the second person formal. So vous parlez would be with an S, and tu parler would be, vous parler would be with a Z, and vous parler would be with an S on the end.
Do you speak French?
Je ne parle pas de français. Un peu. I speak a little of a whole bunch of different things. A lot of very little.
How would you say? How would you say? Do you drink coffee?
Vous. I forget that off the top of my head. Boisson. Vous bois de café.
Okay.
I think that's right.
So when I was in fourth grade, our principal, the principal of our elementary school, who was a sadistic, boy-hating nutcake, psychotic, psycho. I mean, probably a serial killer. He probably has kids buried in his backyard. He decided he was going to teach us all French, and he did it by hitting us over the head with a stick.
That doesn't work well.
No, I didn't learn a lot of French, so every time
I can understand some reasoning behind that, though, because how many times have you heard somebody swear and then say, "Excuse my French"?
Yeah, exactly. Yeah.
See, maybe he just figured if you could get him to start swearing at you, you've learned French.
The other thing about this man is he started out to be a Catholic priest, but they threw him out of the seminary for psychological reasons. So he became our elementary principal.
Wow, that's a low bar.
You gotta love the 50s. 1950s.
That is.
They were good.
I hate to put it so bluntly, but that's a low bar.
Yeah, yeah, it is a low bar. Yeah,
it is a very low bar.
It was too crazy for the Catholics, so they gave him to the elementary school. It's like, and that's probably why we have such.
I don't want to face palm so hard I break my reading glasses.
That's why we have all these screening laws today for daycare and teachers and stuff like that, right? Because of the 50s, right? What they subjected. Okay.
Because of our we have data from the past.
Yes, we do. Okay, so let's end the live stream and go watch Erica and Andrew. We will dig deeper into this. Maybe the Kunin paper. I have a whole huge 2-hour recording of where we went over it, and I think I could just publish that just unedited, and then we could maybe take a look at that and go over that paper again. And start digging into this idea of information, because the creationists are never going to allow us to explain it to them when we are on their channel. Right. They keep coming up with bullshit. So the only way that we can really get into information ourselves and enlighten ourselves is to do it ourselves. So that's what I think. Okay. I'm going to end. So end in broadcast.
Goodbye.
All right.
I want to thank the two of you. And
let me know if you want to continue at some time, if you'd like me to be in on it.
Okay, sure. Yeah. The linguistics part of it is very important, because linguistic evolution has parallels, lots of parallels.
And it's demonstrable, easily demonstrable. So that's important.
Okay. Goodbye, everyone.